So CSCR is a small nonprofit. Here's our webpage. And we engage youth in scientific study of the local watershed and marine environment to inspire stewardship of our planet's ecosystems. The students that we've been serving so far have been high school students and middle school students. If you college students are sort of feeling left out in the dark, come talk to us. And the State of the Harbor is the signature event for CSCR, for the Center for Student Coastal Research. We try to do this annually, and it's an opportunity for our students to present their findings to you, the stakeholders. There may be residents of the town, there may be government officials. We already have heard from Joan Moschino and Patrick O'Connor's office. That's our state representative and our state senator, the ones that work in Beacon Hill and Boston, representing us for the state. We have a lot of other wonderful guests here today. We really enjoy having the diversity of all the people we have here. We have students as young as 6th grade, I believe, and we have some selectmen. We have people who have done a lot for the town. We have residents of Sandy Cove. We have the Gulf Association. We have Tim Davis of the Harbor Committee. We have other people who have done a lot volunteering community service hours for the local communities. We have some leaders in education, and I would say some of those leaders are actually the students. But we also have Patrick Sullivan, the superintendent of schools of Cohasset, and some of our other thought partners that we enjoy working with at CSCR. So thank you everyone for coming and listening to us. I hope you enjoy the show, which is all about the student presentations. So hopefully Beck Labash is ready to share his screen with a video that he has made.
Cohasset, a small coastal town located on the south shore of Massachusetts, is known for its dramatic coastline of exposed bedrock. With many beautiful parks, scenic beaches, and coastal ecosystems, Cohasset's 8,500 citizens spend much of their time outdoors, and often on the water. Within Cohasset's harbor, there are 2 thriving and competitive sailing programs, a maritime center, and a thriving lobster and fishing industry. In the harbor also lies the Center for Student Coastal Research, a place where bright and eager students study the environmental issues facing us today and push for more environmentally friendly practices in our community.
Thank you. Isn't that cool? Beck has his own drone. So our next bit is by the watershed team. They study water quality in our watershed here. Take it away, Greg.
Gentlemen, my name is Gregory Smith, and I am a graduating senior, and I've been part of the Watershed Group for all 4 years of high school. And welcome to the annual 2020 CSCR State of the Harbor event. Before we continue, CSCR would like to say thank you to everyone who was able to make it today, because it demonstrates that you care about our environment as much as we do. This all began 18 years ago with the hope of bringing a greater awareness to our watershed and engaging young minds in the community. As the Watershed Group, we are a family of junior researchers who wanted to bring a rare experience that would unite our community in a collective effort to support our home and fellow inhabitants. We have spent many years working to update officials on the conditions their citizens live in through collecting samples and statistical analysis. I will now happily hand it off to my good friend, Kira.
We were supposed to be on that slide there with Greg, who was just speaking, and now here we go with Kira, who's going to talk about the Boundbrook watershed sites.
So according to the Massachusetts Water Quality Manual, the 6 sites we test at are as follows. Ann River Reservoir and upstream of Beechwood are both Class A. Hunter's Pond near Situate Station, down Beechwood, and O'Donoghue and Jones Street, all Class B. And then a Class A stream or body of water is fresh water and is a designated source of water supply and their tributaries. They are a great habitat for aquatic life, such as fish, and are a great source for primary and secondary contact recreation, even if such use is not allowed. Class B waters are also freshwater and are designated as a habitat for fish and other aquatic life. And then on this graph, you can see the blue line is Class B water, and it goes from Beechwood to North Scituate Station, which is near the Gulf River. And then on this map, you see the same stream from earlier in blue, which again stretches from Hunters Pond to Beechwood. And then you can see that it crosses into both Cohasset and Scituate. So if there's a problem in water quality in this book, it would be a problem for both Cohasset and Scituate. And yep. Thanks. And then on this map, you can see our key sites, starting with Beechwood, which is closest to the fresh drinking water of Erin River Reservoir. Then we have O'Donoghue, which is a new site we just added last year, and it's right off of 3A. After that, there's North Scituate Station, which is by the train tracks right there. And lastly, we have Hunters Pond, which is closest to the brackish water of the Gulf River. And yep.
All
And then
right.
I'll pass it off to Will, which is.
Hello, my name is Will Seavey. I'm an Eagle Scout who's done work to preserve our local environment, and I'm a graduating senior from Cohasset High School. I'm going to talk about some of the lab and field equipment we use at CSCR. We measure tons of things at CSCR, from bacteria to optical brighteners to nitrates, and these measurements help us keep track of local pollutants and where they get into the water. And to measure this stuff, in our lab, you can see in the pictures, we've got things such as incubators, fluorometers, centrifuges, and I'm not going to list all of them off. But when we go out in the field to take the actual samples, there's a whole different set of things that we do, where we take field measurements with a little machine called a YSI, has a little probe on the end. And that allows us to, the one that says screen and probe, we use that to measure temperature, salinity, and other things we wouldn't be able to measure in the lab. And when we take samples to bring back into the lab, we do it with utmost care so no contaminants get into them, such as using gloves. We only use sealed bottles and open them underwater into the stream so no pollutants get in. Or not pollutants, but contaminants. And using all these refined sampling and analyzing methods, we're able to come up with the data that you're about to hear from. And with that, I think I should give it off to Greg again to talk about pH.
Good afternoon, ladies and gentlemen, once again. I'm here to talk about pH, as Will just said. Local waters are often analyzed through an assessment of how acidic or alkaline a substance is. pH is the measurement of the acidity of a liquid on a spectrum from 0 to 14, with substances achieving a measurement less than 7 deemed acidic and those above 7 considered basic. As you'll see on the graph, we had a mostly even year with nothing standing out too much. According to the Massachusetts Department of Environmental Protection, Class B water shall be in the range of 6.5 through 8.3 standard units and not more than 0.5 units outside of the natural background range. As you'll see, located on our map, our northernmost site is Hunters Pond, which we had a little bit of a stick this year. All of the average pH levels for each of the 5 sample sites are considerably lower than last year, with Hunters Pond witnessing the most significant drop of almost 1.5, an entire transition towards acidity in just 1 year from roughly 7.7 to 6.4. Beechwood would end up having the most extreme result with a 6.27. In all, each of the sites had an average pH below the pH standard set by the DEP, though none are more than 0.5 less than 6.5, indicating that all sites are still displaying safe levels of pH. And with that, I'll happily hand it off to my good friend Ava.
Hi, I'm Ava Fusco. I am also a graduating senior, and I'm going to be talking about optical brighteners. So on the graph, you can see there are a bunch of lines. So the red line represents Hunters Pond. The yellow line is North Scituate Station. The purple line is O'Donoghue. The orange line is Erin River Reservoir. The green line is Beechwood. The blue line is Doan Street. So basically, optical brighteners are fluorescent particles found in laundry detergents that are used to make clothes brighter. We kind of use them as an indicator of whether or not septic systems are leaking into our water supply. So the graph right here is the graph in 2018. In 2018, both Hunters Pond and North Situate Station were consistently higher than the rest of the sites, as shown by the red and yellow lines. And then for 2019, the sites Beechwood, Doan Street, and Hunters Pond were relatively low in detergent readings until July 22nd, where they spiked. The site North Situate Station was relatively low until July 15th, where it also spiked. Something that's interesting is that O'Donoghue had high readings for all of the days we monitored it. Additionally, the three highest sites, which were Hunters Pond, North Situate Station, and O'Donoghue, are all connected and close to the Gulf River and Bound Brook, which is an indicator that laundry detergent is finding its way into the water via septic systems based on the spikes shown in the data. And now I'm going to pass it off to Julia.
All right, hi, I'm Julia Gallagher. I'm also a graduating senior, and I've been doing watershed for the past 4 years as well. And at CSCR, we test for E. coli and coliform from our sterile water samples. E. coli is a specific type of fecal bacteria and is one of the most commonly used indicators of human and or animal fecal waste. In the summer of 2019, none of the 5 test sites exceeded the safety limit, which is great. From the summer of 2018 to the summer of 2019, E. coli levels fell drastically at our continuous problem sites, Hunters Pond and North Situate Station, as the E. coli levels exceeded the limit in 2018. Even though the levels were still low, Hunters Pond and North Situate Station still had the highest levels out of all 5 test sites. And the trend line on the graph trends upward as it goes upstream. Coliform bacteria indicates fecal waste contamination. In 2019, coliform levels at Doan Street were continuously higher than the other sites. However, the Beechwood, Hunters Pond, and O'Donoghue test sites all had one testing day with a spike. The presence of fecal waste is an indicator that a possible health hazard exists for people exposed to this water. Here's the 2018 graph for E. coli and rainfall, and the North Situate Station and Hunters Pond, the yellow and the red, are the highest. In 2019, Hunters Pond and North Situate Station were still the highest, as well as the addition of O'Donoghue, which was added this year. Here's the 2018 and 2019 data combined, showing that the 2018 data was much higher at Hunters Pond and North Situate Station than 2019. But the 2 test sites still were the highest, suggesting a problem in this area of the watershed. Now Annabelle will talk about nitrates.
Are you there, Annabelle?
And I'm a graduating senior from Crasset High School, and I've been a part of CSCR for about 4 years. And I'll be talking about nitrates. Nitrates are a source of food for algae. When nitrates are in excess, an overabundance of algae may occur, choking out the aquatic life by depleting oxygen in a process known as eutrophication. An increase in nitrates is often caused by human pollution, with the most common cases being sewage system leakage into surrounding waters and excessive use of fertilizer. As evidenced by the 2018 and 2019 data, nitrates have been consistently high at North Situate Station and Hunters Pond throughout the years we've been testing for it. So on the graph, the red bar is Hunters Pond and the yellow bar is North Situate Station. And even though they get lower in 2019, the red and yellow bars are still the highest out of all of the other test sites. We're wondering if this could be the result of sewage system leakage and runoff from fertilizer. It's also important to note that North Situate Station and Hunters Pond are in a shared watershed that occupies both Crasset and Situate. And I'll pass it off to Alex now.
And the other thing that I think that Annabelle was going to talk about is the idea that it could be evidence, though we're not certain, but it certainly could suggest that there's septic systems that are leaking into the water. And so now it is Alex's turn. So Alex, why don't you jump right in there?
Hi, I'm Alex, and I'm another senior at Cohasset High School. I've also been doing CSCR for the past 4 years, and I'm going to talk to you about chlorophyll A. We use a fluorometer to test for chlorophyll A, which is the green pigment found in plants, to determine the presence of macroorganisms. First, we filter the sample water, then we dissolve the filter paper in a concentration of 90% acetone, we let it sit, and then finally we use the fluorometer to read the chlorophyll levels. Surface water with high chlorophyll is usually high in nutrients, specifically nitrogen and phosphorus. These nutrients can cause algal blooms that deplete oxygen, which is deadly for fish. We'll just mention this process a minute ago, talking about nitrates. High levels of nitrogen and phosphorus can also signify the presence of anthropogenic pollution, such as septic leakage, poor water treatment, and fertilizer runoff, which you also just mentioned. Therefore, we can use chlorophyll measurements to determine both plant growth and pollution. During 2019, chlorophyll A readings pretty much aligned with our expectations. The highest levels of chlorophyll A were found at the Doane Street site, followed by Beechwood, O'Donoghue, North Scituate Station, and Hunter's Pond. Those sites are listed in order from freshwater to saltwater, with the most chlorophyll A levels being inland and upstream. It's interesting to note that this is the exact opposite trend for optical brighteners, bacteria, and nitrates. It would seem that plant growth is greater where there's less pollution, but we can't draw conclusions yet. This is definitely something Watershed's going to want to look into more in the future. Now I'm going to pass it off to Andre.
Got it.
All right. Okay. Sounds good. Alrighty. So I was tasked with discussing dissolved oxygen, which is very important for fish life in these general areas. And what dissolved oxygen is it's the amount of oxygen gas that's been saturated into the water. And it's affected by a bunch of things, such as salinity and temperature. Sorry, my wire's going insane. And so our main sites that were at an acceptable level were Hunter's Pond, North Scituate Station, and Doane Street, all of which are running water sources. And the two sources that we saw were actually problem sites were Beechwood and somewhat O'Donoghue, which are very stagnant and slow moving. Beechwood way more than O'Donoghue. And so this just kind of gives you a good idea of how much dissolved oxygen, which my previous chart showed. Right here is 5 milligrams per liter, 10, 15. And this kind of shows you just general fish life that can survive at those levels. And most sites on average were right around the 5 to 6 range. And Beechwood tended to stay around 3 or 4 to 2. And I discussed very briefly a little minute ago what affects dissolved oxygen. And those two things are salinity and temperature. And the way that works is as temperature or salinity increases, on average, the dissolved oxygen milligrams per liter decreases, as displayed by this chart. So as you can see, salinity, as it's increasing, the dissolved oxygen decreases. And as temperature increases, so the dissolved oxygen also decreases.
Let me just say one quick thing to the audience before I hand it over to Susan and her team. These guys did a great job in talking about all this data, putting this together. This was their research. These are their graphs. We helped put it together in a story map format. A little shout out to Mike Thompson, who's here, who is a GIS expert from Stellwagen Bank. But I just want to go back and say 30 seconds worth of talk about the map that Kira mentioned, and I'll stop screen sharing. So one thing that we didn't say is that for years, and Doc T's here with us too, and she can tell you, for years we've been testing the Gulf River, and then we kept thinking that there's something going on in North Scituate, and that's where the watershed team took over. very back upstream. And at one point we were all the way up here testing, and then we kind of focused on this part of North Situate. And as Alex said eloquently, and Julia and Ava, et cetera, we and Annabelle with the nitrates, we kind of think something's going on right there in the North Situate area, and the investigation continues. So that's the watershed group, and I will turn that over to Susan and the eelgrass team, who are going to take us now downstream, down the Gulf River, out into the estuary of Cohasset Harbor.
All right, Andrew, are you ready? Excellent. Okay, eelgrass team, thank you.
Hi, I'm Lucy. I'm a junior, so I'll just be going over this. So this is eelgrass. So in short, it's the seagrass, so it's much longer than normal grass and it's underwater. So this is a screenshot of our Survey 123. So basically our Survey 123 is a ArcGIS collector that we use, and we just switched to Survey 123 in the summer of 2019, I believe. So this is a screenshot of what it looks like on our phones when we're out on the boat. So we can put in certain observations such as the eelgrass coverage density, the bottom or sediment type, or the motile organisms, and any other observations that we come across. So this is basically how our Survey 123 data transfers onto our ArcGIS. So we can use different attributes to show different things. So we have 3 different layers shown here. As you can see, the legend says that one is coverage density from data up until 2019. And then the other one is coverage density from 2019 on. And then the third one is sediment slash bottom type from data up until 2019. And so as you can see, that circled area, that is a newly discovered area from this past summer. And it was discovered by our fellow researcher, Connie Chapman. We decided to name it after her, so Chapman Meadows is what we call it. And now Andrew's gonna talk about some orientation points for our map.
Yeah, so some reference points for all of this data that's just shoved in front of you is right here is Whitehead, right over here is Sandy Cove, this area over here is the Glades, and right here is the Adam Residency.
Okay, so now we're gonna show you some footage that we have. So we get a lot of GoPro footage, but we thought that this one was especially special because you can see a fish. And typically what happens is we deploy the GoPro and it's sitting on a plastic quadrat, and we put it in the water, but we also usually have swimmers or people swimming around to take samples. So then when we take samples, we look for reproductive shoots, we look for leaf length, and we look for tunicates and other organisms. And yeah, so this is a good example of one of the shots that we have. So yeah. There's a fish. And we have a lot of footage, so we had to find the special one. Okay, so next, turbidity. So turbidity is basically how clear the water is. So we found that from our data from 2019 with our ArcGIS data, the visibility ranges from about 1.5 meters to 6 meters in our harbor. So that picture right there, that's a Secchi disk, and that's how we measure turbidity. And it matters because the more turbid the water, that means that our eelgrass can't photosynthesize as well. So it's an issue. I'll pass it off to Alex now.
Right, thank you, Lucy. The reason we care about things like how well eelgrass photosynthesizes because of the water turbidity is because the plant plays a role in the ocean's function as a blue carbon sink, mitigating the effects of climate change. Eelgrass absorption of carbon also relates directly to its ocean acidification, which determines the ability of shellfish to grow their shells, which in turn is going to affect shellfish aquaculture. So eelgrass is really important and affects like a lot of different things. Next slide, please. Meanwhile, on a more local scale, a variety of species from mammals to fish to waterfowl and crustaceans rely upon eelgrass for food or shelter. Eelgrass is a primary producer in the food chain, so it's like, can't live without it. And some of these photos I got on the internet, and then a lot of them are like actually taken from eelgrass in the field. So those are like, a lot of those photos are in Cohasset of animals here. The town budget also relies on eelgrass unexpectedly. So nor'easter storms naturally push sediments southwest towards the canal. Andrew, will you circle the canal really quickly just so they can see where it is? Yeah. So where you see all the eelgrass on this map is like, it's holding all the sediments in place. And so if it weren't doing that, if the nor'easter storms were pushing all the sediments southwest into that canal, the canal would need to be dredged a lot more often than it actually does. And this would be very expensive for the town. Next slide, please. So by preventing environmental changes, filtering the water, and feeding and providing holds for species such as oysters or blue mussels, eelgrass feeds our economy as well as the coastal ecosystem. Lobster fishing, and shellfish harvesting rely on a robust and healthy food chain, of which eelgrass is a linchpin. All right, one more slide. Therefore, finding ways and times of days to access the upcoming shellfish aquaculture project without harming or disturbing the eelgrass in any way is really important. It should be on the top of any shellfish farmer's list. And now I will pass it off to Andrew.
Hello, I'm Andrew, and I'm a junior, and I'll be talking about what we're doing now. With the restrictions put on us now preventing us from collecting data, we have been using the time to review GoPro footage, as shown before, and figure out which videos contain important data. Along with reviewing GoPro footage, we also participated in the online Zosterapalooza, where eelgrass experts presented their findings of the past year. Our biggest development in the past month has been our application for the Margeau Foundation Sea Grant. With this grant, we plan to take our process of data collection to the next level, where we apply what is learned in our classrooms, such as statistics, to achieve an accurate model of the eelgrass in our harbor. And I will now pass it on to the drifters.
Share my screen.
Yeah, awesome work, Lucy and Alex and Andrew. And if Susan didn't say that,
if you want, I have it up.
Beck is now going.
Beck first and then drifters.
Yeah.
Oh.
Yeah. Beck, are you ready? Here we go.
Despite our town's progressive attitude towards the environment and local ecosystems, Cohasset's beaches are continually plagued by one thing: trash.
The trash that litters the shore of Cohasset and every other coastal town has been given the name marine debris by the researchers studying it. According to the National Oceanic and Atmospheric Administration, or NOAA, marine debris is any persistent solid material that is manufactured or processed and directly or indirectly, intentionally or unintentionally, disposed of or abandoned into the marine environment. We know the definition of marine debris, but how do CSCR students perceive marine debris?
Anything man-made in the ocean that's not supposed to be there. Human manufactured items that enter the marine environment.
Trash that is floating in the ocean or around the ocean.
What should be done about marine debris?
Watching what we throw away and cleaning it up if we see it. Besides just cleaning up marine debris that's already on our beaches, we can make sure we're putting trash and recycling in the proper places.
Community beach cleanups are a good way to solve what's already there, but recycling can prevent it.
Not littering, and if you see any plastic anywhere, you should pick it up.
Over the past few years, a group of students from the Cohasset Center for Student Coastal Research, CSCR, have been studying marine debris to try and understand local marine debris and draw conclusions from our data. We have conducted cleanups and gathered data from various beaches around Scott's Shore: Sandy Beach, Brush Island, Rocky Beach, and Bassings Beach. Some of the hypotheses we've been investigating are:
Number 1, does marine debris come primarily from land or sea? We surveyed both private and public access beaches and found that they both had very similar percentage of debris composition and the types of plastic. Food wrappers, straws, fishing gear, etc. were consistent for all areas surveyed. Number 2, does the composition of the debris determine where it will be found? We found that floating debris is commonly found higher up on the beach, while denser materials like glass and plastic collect more towards the water's edge. Number 3, is there a certain type of debris that is more prevalent on our shores? We found that plastic makes up the majority of marine debris found during our surveys and cleanups. Fishing gear also made up a large percentage of our local debris. One location we investigated was Brush Island, a small rocky island off the coast of Cohasset that acts as a nesting location for gulls and cormorants. We found the island to be covered by dozens of derelict lobster traps and hundreds of feet of rope.
Based on NOAA procedures, we conducted an accumulation survey on Bassings Beach in Cohasset. We chose a 100-meter section of beach and surveyed and collected all the marine debris between the water's edge and the vegetation at the top of the beach. We gathered data using an app developed by team member Beck Labash to categorize different types of debris and create GIS maps of the data included. A shoreline survey is more than just a beach cleanup. We do remove all the debris we find, but we are also trying to gather data about the types of materials we find and draw conclusions.
Marine debris is a global problem affecting every coastal location on Earth, including Cohasset, Massachusetts. Marine debris pollutes the environment, concentrates pollutants, and can kill wildlife. Plastics emit deadly toxins when they break down into microplastics. Microplastics have been detected in the seafood we eat, the water we swim in, and the sand of the beaches we enjoy visiting. There have been many attempts to reduce and prevent the contamination of the environment, such as phasing out plastic for more biodegradable materials, increased enforcement of existing laws about littering, and increased public education about the issue. Many local groups have also begun annual beach and oceanic cleanups to help get rid of the already existing problem. CSCR hopes to continue to study the problem and aid other researchers in their studies as to how best to prevent the destruction of our Earth by plastic.
Hey, Susan, if I can just jump in right there, I want to let everybody know, obviously those, you can see the chat yourself, and there's some accolades going around. So we've been looking at all of this material together over the last week, and that was actually the first time that all of us have seen the second half of Beck's video. So we saw the first half, the intro, and that was queued up in the beginning of this presentation. But that second part that he just played is the first time that we've all seen his final product. And man, was that a home run? That was a grand slam. That was a beautiful piece of art and science put together, Beck. So just wanted to give that little shout out before Susan takes us through the next transition.
So the next transition is pretty easy. That was awesome. And the next one is the Drifter team. Sounds like a strange name, but they'll explain what they mean by that. Are we ready, Carl and Andre?
Yep. Go share it, I guess. Okay, Drifters, current in Cohasset Harbor. Drifters is a program where we build drifters, which are pretty much our devices that track ocean currents. The frame of drifters are made out of PVC piping, and we use canvas sails to catch the currents underneath the water. The bottles on top, as you see here, are they track GPS trackers that pretty much tracks location, position, and the direction of the drifters.
One sec,
So
Carl.
here you can see drifter sails submerged underwater, and these sails right here, they pretty much catch shallow ocean currents, and they propel the drifters around. So to deploy the drifters, we use a canoe and kayaks to go to Cohasset Harbor, and then we drop the drifters in predetermined locations. We let them move for about 2 hours, and then we retrieve them and bring them back to CSCR building. We then transfer the data from the GPS trackers to the computer, and we compile the data into a map. So you see here is the raw data from the drifters, and you have the date, the time, the latitude, longitude, the multitude radians and radians. Then we have the distance, the time, and the velocity. Here. And we pretty much take this data, compile it into a ArcGIS format, and create a map. The map looks like this. So as you see here, blue means the fastest, and red means the slowest, and the direction shows the position of the drifters. So as you see here, this is a Gulf Stream current that sweeps from it sweeps through the Cohasset Harbor, causing fresh water into the ocean. Here is a eddy current, which is a current that swirls in a circle. And here we deployed some drifters to track the water treatment plant. Anything on that, Andre?
Yeah, so the one that he just pointed out, that's located right off the docks of Atlantica. We usually put around 2 or 3 there. Then the Gulf Stream is always usually one of the fastest because it's pulling it out into the ocean. And yeah, that eddy current, that's off of Bassings Beach. And not much else I can say. You did a pretty good job.
That's great. I can't resist jumping in again. Carl and Andre have recently worked together to put that together. And Carl is going to knock it out of the park this summer, spring and summer and fall and years to come, because he's really interested in crunching that data. So it collects a massive data sheet, and one of the teachers in the picture, Scott Leitz, is a math teacher who was working with students to turn those GPS positions in time into vector velocities that would show speed and direction. So it's quite an awful lot of work just for what they did. And they did a great job putting that together. So thank you, Carl, and thank you, Andre, for that. Sorry to keep jumping in, Susan, just can't resist. Go ahead.
No, that's good. I'm so proud of everybody. It's great. So the next project is actually my project. My video editing is not as good as Beck's, but I have a problem because I've got these great junior researchers from 5th to 8th grade. I love them so much. They're great. And the reason that we're doing a video is if I let them talk, they have so much information, they would go on and on. So we had to restrain them into a video, but they all contributed, and here it is.
Bassings Beach is an island off the coast of Cohasset Harbor. There are 3 ecosystems that the CSCR team is studying: dune vegetation, salt marshes, and shellfish. Today, my team and I will be describing why they're important and why we should care.
It's kind of like a sentimental thing. Since the summer research, we found it, and it's a good spot to like meet on and that's where a lot of research, such as the goo project, which is just a weird substance on Bassings Beach, not solved yet. And there's a lot of dune grass projects that just happen in that general area. So we just mark it as a waiting point. We actually have it marked on ArcGIS, which is a program where you mark on pinpoint geographic points on latitude and longitude.
As I walk around the vegetation line, I use a tracking finger to measure the distance and direction around Bassings Beach.
We want to track the vegetation line for the Ammophila, so that we know where it is, because it could change over time, and we want to know where it is now compared to the near future, and in that way, in a way, we can see whether the island is changing or shrinking in any way.
What's Ammophila?
Ammophila breviligulata, also known as dune grass, is basically all that green stuff there.
Wind erosion is basically erosion with wind. So if you were in a desert and you see a bunch of dunes, that's caused because of the wind blowing them.
Bassings Beach, it's pretty flat, and then there's a little slope right up to some of the plants. So on a windy day, just even one grain of sand would be flying around.
It looks pretty chaotic. Most of the plants have pretty deep roots, like the Ammophila.
Yeah, the stems of the plants can catch the sand.
There was recently some strong wind in one of our subtropical storms.
We had a king tide,
So Nick just paced it out, and he said there were 10 paces between where you're standing and where the water came from the ocean side, all the way to the tide line that came from the upland side.
Oh yeah. We made the rocks go in like 1 square meter around the plant.
Can you tell if it's holding any of the sand?
I believe it is. We could dig it a bit. It seems to have a firm rooting system.
And you can notice how when the wind comes, it doesn't rattle this plant, but it rattles the reeds right there.
Here I have a chunk of Spartan altum flora growing in this peat with this slippery substance on top of it. And you can actually see how the roots grow, that's what holds the peat together, all the roots in it. And you have rhizomes going off here, not fully yet extracted. It's just the tail of the rhizome. And you have more roots going through here, and then this is the hole it's in. So you can see the ground I got it in. Nice. That's where it is. It's really cool how it's like that.
Where did that marsh go? That was here somewhere. Wasn't there a marsh somewhere?
You may notice that this line lines directly up to that line. The peat seemed to have existed here before the sand. It is directly over the lines of the peat. And if you notice, there was once marsh grass here because you can see the stems. They're still holding it together, and there is still there. And you can see it is slowly dying out as the sand piles over it, which almost seems that it was almost all blown here from somewhere else. So maybe the sand is really just over the peat. We may notice that they're at the exact same altitude, which we can furthermore test. Thank you. Hey guys, we're back at it again. Today in the CSCR, we went to the cove in Cohasset and we looked for wildlife. Today I learned that clamworms have teeth and fight each other. Oh, serious jam.
I don't know who's winning.
I think that guy's winning right there.
Today I identified worms in the lab.
What do we think it is?
We think it's a anopheropod.
Say that again?
It's an anopheropod.
And that sounds like you're making up two things. Is it right here in this book?
Oh yeah, it is. Yeah, it's annelid. It's a phylum.
Phylum?
Phylum annelid.
Yep. Great. Beyond the breakwater on the north side of Bassings, we are particularly interested in where the shellfish are. This is a map by Morris, available online, and this is our map showing the low tide line that we actually walked and the shellfish that we found. This is the map from the Scituate Shellfish Advisory Commission proposing their 8 pilot project plots, and we like to provide you with maps and visuals so that you know which areas they're talking about so that accurate decisions can be made. 2 of our students have written a grant application to keep researching in this area and keep you updated on developments.
Yeah, we definitely could lose Bastings Beach and other places like it.
People in Cohasset should care because this is a coastal town and some of our marketing relies on the coast.
Oh, it's not as steep as it used to be. The sand is getting washed away by the ocean.
How long ago were you here when it was noticeably steeper?
2 weeks ago.
2 weeks ago.
I think it was 2 weeks ago.
Alright, thanks guys for watching. Just remember to like and subscribe and hit that notification bell to get sure when that next video pops out. See you later!
All right. Yeah, that's
You know, when Noah said it's really cool, it is really cool. And it's, I want to give a shout out to Susan Bryant, who does tremendous work with that group of students. And Susan loves to talk to the kids about having curious questions. And Susan and the students are able to really identify some curious questions that they're interested in pursuing. And don't they really just literally dive right into it? And it's just great to see. And they got that cool stuff going on, like the GOO project, where they're out there and they're looking. Their curious question about what's out there is, what is that goo? What is that oily, slimy substance that seems to be pretty prevalent in some low-lying areas out there? But tons of awesome questions, and great job, Susan.
All right, so our next part is the other projects.
Yeah, and I'm going to be really quick here. Yeah, so I'm going to go with the screen share and just add it to the end of this story map are a couple of things. And Kira, if you can hear me, and you're there, and Percy, but Kira put this together that is about, let's see, where are we? There's the drifters, and two things to mention: Cohasset beaches, the monitoring work that we do with the town of Cohasset, Treats Pond, and Sandy Cove. So Kira, are you there?
I am, yes.
Yeah, so Kira helped.
I'm not muted.
What's that?
Nothing, sorry.
Okay. So for everybody listening and viewing, this is not something that we've rehearsed. That's why I'm giving Kira the shout out. But Kira's the one who put together this, this data. So here's Black Rock Beach, or Rocky Beach. This is Cohasset Sandy Beach, public beach. And here's Bassing's, right across the harbor. Everybody knows the popularity of Bassing's Beach. The one thing that I want to do here in this slide is give a shout out to all the students who do the bacterial work at CSCR. So we have the honor and pleasure of working with Pam Fahey, who is in the audience today, and Pam is Town of Cohasset Health Agent. And we collect samples with Pam for the Town of Cohasset, and they get delivered to GNL Professional Lab in Quincy, and we run samples ourselves. We're able to return the data to Pam before GNL is able to return their data. Ours comes early in the morning, 24 hours after the sample's been drawn. Theirs comes 24 hours after they've run the sample and get time to report that out, which is usually 3 or 4 in the afternoon. And our data tracks awesomely is not the word I want. It is, it meets all data quality standards. And there are, on this particular graph right here, where you can see the 3 red, they're just over 30% relative percent difference log of the relative. All of the others are well within this band of being spot on with the professional lab. So we still don't know why, back to the map, I'll go to the map again. We still don't know why all the time we tend to get closures at Bassing's Beach and Sandy Beach. It's definitely a mystery. And the other thing that we've been doing is investigating Treats Pond and Sandy Cove. And Treats Pond is a long studied issue in the town of Cohasset that has drainage to Sandy Cove, and the drainage gets blocked by the accreting forces of the beach here. And it needs, so the question always is, should Treats Pond drain out? And Percy Hillier, who did a lot of work this past fall, put this together. And the takeaway here is that the bacteria counts in Treats Pond are incredibly high, and Sandy Cove tends to be very, very clean. Occasionally you might get a high reading depending on seasonal variety, but for the most part, this beach area is pretty clean in bacteria counts. Very little concern. Whereas Treats Pond is the epitome of a petri dish. And so that sums up the bacteria work that we've done at the beaches. Kira's been awesome in collecting that, and Percy gets a shout out there. And there's a bunch of students who have been involved in that program as well, and a thanks to Pam for the work with her. But I want to give the some audience members first shot at any kind of remarks that they would like to make. So I might, forgive me if I miss some of the scientists in the audience. I don't know if our board member Karsten Haber's still with us, but Dr. Haber and Dr. Ann Tomei, board members, if they want to give a comment or two. Mike Thompson, who is a GIS marine biologist specialist at Stellwagen Bank, Jill Carr and Sarah Grady from the Mass Bays, Carolina Bastistas from MIT Grant, Sue Flint I know is here from Mass DEP, Pam Fahey from, as I said, Town of Cohasset, Jason Bertner, Coastal Zone Management. So we have a lot of expertise in the audience. So forgive me if I didn't just give you the shout out.
Don, former student, Don Massa of Massa Products on 3A in Hingham.
Yeah, we do have some former students. I think I got a text from Dawn saying she was having trouble logging on. And I don't mean to overlook anybody. But I do want to sort of throw it out to say some of the policymakers, because here's the reason why I'm doing it this way. Originally, if we were all meeting in Wilka Commons as we had planned, we would have had some of these names that people I just mentioned. We would have invited you to sit as panelists. And the question that we have for our students, the question for our policymakers and scientists at the local, state, and federal level is who uses our data? And for the students, we always tell the students that the data that you are presenting is badly needed by different stakeholder groups. So how are our findings used? Thank you, Susan, for throwing that up there on the screen. And in no particular order, I've already said too much, in no particular order, if Jill or Sarah or Carolina, et cetera, want to jump in and say something, please do.
Okay, can you hear me?
Yep.
Great. This was really fantastic. So I applaud all of the students and anybody who assisted them and guided them along the way, especially Susan and Jack. This was really great. A lot of the data that you're collecting can go straight into programs that state agencies and other researchers are working on. So for example, I've worked with some of your eelgrass data in the past. Your presence absence information about where eelgrass exists and where it doesn't is so useful. You can pass that on to the Division of Marine Fisheries and Mass DEP to incorporate into their own mapping programs, their statewide mapping programs. And of course, your water quality data is already used by Mass DEP in their impairment listings and water quality monitoring. So you guys are really making the connections that you need to make your data useful and relevant. And I think you're doing a great job with that.
All right, I'm gonna stay in teacher mode. Jason, you're up with Sue Flint on deck and Doc T in the hole.
Hi, one of the things that I'd like to say is in addition to the data itself, I think one of the really important functions that CSCR and the student educators have is just simply public involvement and bringing these issues to the public and providing forums such as this to help present out on that information and discuss the ramifications of this type of studies, these type of data to the communities and help how that can inform public policy on the local level. And again, great job to everybody involved. This was a wonderful presentation.
Thank you. I forget who I said, so I'm going to go with Ann. And I know Pam wants to talk and Joan wants to talk. So Doc T.
Oh, good. Yeah, I mean, how many accolades can we give? But this was just such a fabulous presentation. I learned so much more. I'm highly involved with the Gulf River Watershed Water Quality Group, but learning about what the junior marsh monitors are doing and the drifters and the beach bacteria testing, I'm totally blown away, and the eelgrass, and it's fabulous. I don't really have any general questions. I'm thinking about our project for this summer and what that's going to look like. And basically, I would want to ask the watershed group if they had to whittle down their like hotspots and what they really want to test. I saw that Steve Wenner asked a question about why the pH levels dropped this year compared to last year. I had a question about the bacteria levels being less in 2019 than they were in 2018. This is for the Gulf River Watershed project. And I don't know that this is the right forum for that. I feel like I just would rather work with the 8 or 10 students and kind of narrow down the focus for this year. But thank you. It's amazing. Thank you so much.
Pam, I know that you had your hand up. Pam Fahey from Cohasset Board of Health.
Oh, thanks, Jack. I just want to say, first of all, these presentations were great. And I really wish there was something like this when I was a kid. I was an environmental science major and I would have loved this kind of thing. But the beach bacteria testing has been a huge help, like you mentioned. You guys give me the heads up. Like you said, I get the results before I get the results from GNL. So I know if, say, Sandy's got a really high level coming, I can run out, grab a sample, drive it up to Quincy to GNL before the end of the day. Otherwise, I'd have to wait another day. And if we have to close the beach, it actually buys us an extra day to get the beach back open again. And then 2 years ago, not this past summer, but the summer before, we were getting really high hits at Sandy. We couldn't figure out why. And I didn't have it in the budget to keep taking additional tests. So CSCR stepped up and really helped me out and took additional samples. Instead of just taking one at Sandy, they were taking like 3 across. And we were thinking maybe there was a problem near it was the same summer that the new bathhouse opened up. We were thinking maybe there was a problem with that. And we kept getting high hits thanks to the CSCR results down the complete opposite end, which didn't make sense because we thought everybody was on sewer. We found out once the season ended that there was a house there that had a septic system that they didn't tell anybody about. And even though they were on sewer, it was the septic, and the CSCR results kept pointing to that. And we were like, no, that doesn't make any sense. It can't be true. And it turned out CSCR results were pointing to the problem, and it ended up getting fixed. So last year, we ended up with no issues there in Sandy Beach. So thanks to CSCR, helped solve a mystery, and I'm going to keep using you guys. You guys are great.
Oh, thanks so much for that anecdote and the shout out. That's awesome. So Joe Moschino, I see you have your hand up, and then Dawn Massa Williams Stankovic wants to speak as well. And I'll give Dawn a little intro after Joan speaks. Go ahead, Joan.
So first of all, this is, as usual, fantastic. I loved the presentations. You guys are just tremendous, but I wouldn't expect anything less. And I'm glad we were able to still do this by Zoom. I wanted to go back to Lucy Gray and eelgrass. And I would just love if you guys, no rush, we're sort of coming into the end of the legislative session, but you did ask about how can some of your research be used. And as you all know, I'm very much, I filed a big climate change bill. I'm supporting bills that relate to sustainability and really the decarbonization. And Lucy talked a little bit about how eelgrass functions as a carbon sink. And I would love to have just a modicum more information around that because there's actually a really interesting bill that was filed by one of my colleagues on the Cape, Sarah Peak. And it has to do with preserving our carbon sinks. So think about the forests. Massachusetts has some of the oldest forests in New England, and those function as a carbon sink. But then we also have all of the coastal communities and all of like the marshes and those kinds of things also reportedly function as coastal sinks. And so I'd just love to see a little bit more of that information if you guys didn't mind. No rush, and I'm not giving anyone homework. But so at some point, I'd love to either have a little bit more of a detailed briefing on that, and we can elevate that in, you know, in the remarks that I make that support that bill, and in partnering with Rep. Peak on how she advocates for moving that forward, whether it ends up being this session or next session. So I'd love to partner with you on using your data to help my advocacy for our community.
That's awesome. Thanks so much for that opportunity, Joan. And it's you know, we often like to remind the students that there are people in the real world who really want to know what results you're finding. And it's just great to have that affirmation, that confirmation, that validation of the student work. So thanks so much for that.
Yeah, and I listen to everything you say, and I made notes for myself on where are the potential spaces. Not everything is a legislative solution, but I did listen for everything you're talking about and I'm sure Anthony from Senator O'Connor's office was listening with this too, about what, if any, legislative solutions there are, or what, if any, high-level state policies that might lead us to. So keep up the good work, and keep, especially around the sustainability and the healthy ecosystems, and just keep feeding that to us and keep us engaged on that, because we might write a bill based on your work. You never know.
That's awesome. We just need to get that line item into Charlie Baker's budget that CSCR gets a couple of a one with a lot of zeros following it.
Working on it.
But anyways, I see Sue has her hand up, and I said also that Sue Flint from DEP. Sue, do you want to speak?
Sure. This was great. I was very impressed with all of the presentations, and especially all of your work with sort of the newer technology, the GIS and you know, ARC survey and the GoPro footage. That was all fantastic, and that's stuff that will serve you well going forward. Yes, DEP, definitely we use the data that we can get. We have a strong team of people who do field work all summer, taking data in rivers and streams and coastal areas, but we can't be everywhere. So we really love to have the extra data to do the assessments. And by assessment, I mean, so you said early on, you know, you gave Class A and Class B, hopefully no Class C waters in your area. But the assessments are where we look at the water quality that would support those goals, those uses, and we try and figure out whether they're meeting the uses. So it's an important piece of information. We were really happy to be able to give out the bacterial monitoring grants this last 2 years now. And really happy to see the quality assurance. I'm the quality assurance person for our program. And just, even though it seems maybe like a waste of time when you're going out and you're taking 2 of the same samples in the same place at the same time, that's the information that's really critical and really gives credibility to the data that you're taking. And, you know, informs you whether it worked out well or not. The one thing I wanted to jump back to the pH and the differences between last year, 2019, 2018. Excuse me. I think that would be a super interesting project for the stay-at-home time, to take a look at all of the information you've got. So look at your calibrations on the instruments, look at the time of day that you took pH, because that does vary over time of day, especially in a really eutrophied system. And take a look and, you know, lay it upside with some more general weather data, and see if you can tease out where that really kind of big jump was in pH. And maybe if you have other years of data, it would be super worthwhile putting that up. Great presentations, thank you.
Thanks so much. Yeah, we do. We didn't have the bandwidth, as they say, to put it all together, but we do. We have some good 2016, 17, 18, 19 data but the format wasn't conducive to putting it together for this particular presentation. And the DEP monitoring of the Weymouth Back River and Ware River and Musquashkett Pond, we couldn't connect with the students who were doing that work, so we didn't present that data. Otherwise, we would have. So next year we'll have that for you, Sue, and we'll be back with Carolina. I see you on my screen. So just a quick opportunity if you want to speak. And Diane Kennedy, I know you're there, and you might want to say something. And Ed Savage, and here's Dawn. So Carolina, why don't you just go ahead, and then Dawn's on deck.
Sure, thank you for the opportunity. It's great work that everybody's doing there. Is well, besides having a lot of fun that I'm sure you have, you are really producing like something that I would say that is a gold standard for every scientist, right? Is to have a data set that will help interpret what is going on in the community. So for example, the water quality data that you have together with the maps of the eelgrass and how is the density and how the shoreline is moving and so on can help interpret many things. For example, I work specifically or try to work with ocean acidification vulnerabilities, and it's impossible to try to interpret the coastal values of pH if we don't have a lot of information that you already have. So for example, to interpret how much will be vulnerable a particular shellfish, it's also important to know how much nutrients you have, how much variation you have in salinity, and so on. So all these variables that you're collecting are really gold standards for many of the implications that your work that climate change could have in these communities. So I think it's a great value. I congratulate you guys and keep the good work.
Dawn texted me. Dawn, I knew Dawn as Dawn Massa Williams. And Dawn, forgive me, I don't know if you pronounce your name Stankovish or Stankavish or some other combination of that, her married name now. But Dawn Massa Williams was a student of mine a while ago. So she's been around since we've been, since the alternative school existed at Cohasset High School. And I'll let Dawn introduce herself in her work that she does at Massa Products on 3A. You've probably driven by Massa Products on 3A if you're from this area many times. And Susan and I, I'll say one quick thing. Susan and I had an opportunity to meet with Dawn and her dad and a couple of their engineers, and we got a private tour of the plant. And if Dawn's ever offering tours, that is a must-see place. It is unbelievable, the facility that they have there. So, Dawn, are you there?
I'm stuck having to call in because the DoD doesn't let us use Zoom, because at Massa we design, we engineer, and we manufacture sonar for the Navy, as well as ultrasonic products for and in air and in water and in fluid products for industry as well. And yeah, I thank you, Jack, for the introduction. And I wanted to say firstly that I'm very impressed with all of the students. I only wish I could have seen all of the wonderful videos, but it was so well described. I feel like I was able to see exactly where we were in all those maps, because I know the area so well. In fact, when I was a student with Jack, I was taking some samples in some of those same locations. So it brought me right back to that. And there's so many elements of what the kids are doing now that are so important in their life. What you're learning is going to stay with you, as it did with me. It'll influence you and inspire you to do wonderful things in your life. And I'm very excited now in what I'm doing to be able to figure out ways that we can enhance the data collection that the kids are taking, and enhance the quality of what some of that data might be, and put it all together in ways that could really help everybody achieve multiple goals from all the different ways that you could go with this. Everything from the marine biological standpoint of just the ecosystem there, all the way to what's going on in the waters, to how it's affecting other life forms, from something small as bacteria to as large as whales potentially. So there's a lot of exciting possibilities that I see that we can talk about and figure out where we could go. And it's the kind of thing that totally goes full circle, and it's the best kind of learning possible.
Thanks so much, Dawn. And it's great to reconnect with Dawn, and we're looking forward to working with Dawn and her colleagues to, as she had suggested, work together to provide the students with some enhanced capabilities for their studies. So we batted around some ideas about trying to do some bathymetry with some of the sonar equipment that they've developed. So that sounds way cool. So when we get out in a boat together, we'll invite you all along. I don't know if Diane Kennedy, are you there? Did you want to say anything? I don't know if she's muted, but Diane is a...
Hello, I'm just going to say hi, and this was terrific. I just stepped away for about 10 minutes. Thank you.
Diane is one of our select board people in Cohasset. I forget what word we're using now, sorry. And has served for many years in that role, and has been a great supporter of CSCR, so we appreciate that. I want to just jump to talking about our role in education. The work that the students have done, and let's keep in mind that these are middle school students and high school students. The work that they've done doesn't come with a test at the end of the year. There's no standardized tests. All of this learning is things that they have, is them learning what they want to learn. It's motivated by their own curiosity and participation in a team. And in education, education is full of jargon, and the word that we use for that is authentic assessment. So you all, as our audience for this, are giving the students an authentic assessment for the work that they've done, and your honest questions are what makes this real and makes it really exciting to do education the way that we do it here at CSCR.
And thanks, Susan. And I can't thank you enough for being with us today. We had 97 and a few extra after that noon cutoff. We had 97 people register. I know we had as many as 80 on at one time, and this was phenomenal. And just to pick up with what Susan was saying, I'd like to give a shout out to the students.
See, if you didn't get to your question, or if you come up with one afterwards, we love questions. Curious questions from students are great, and curious questions from adults are great. There's our email. Please email us. Or you can find it on our website, which has an extra C for Cohasset, CSCR.
It's not the best shout out for all the students because I don't have a picture of everybody loaded, ready to go. But I wanted to give a shout out to the seniors, and I know I'll make a mistake by forgetting somebody, but I'll try not to. But Greg and Annabelle and Kira, now Andre's not a senior, that's why I skipped him there, and Julia and Greg again, and here's Andrew and Lucy and Susan, not a senior, working together, basically working on this report way back in the winter, and Kira again, and Catherine, who isn't with us, and Ava, and Will with the sun in his eyes, and Alex is not in this picture, but Alex featured prominently in the earlier picture next to the automated burette. There she is. There's Alex doing the chlorophyll A work. And so I just wanted to give a shout out to some of the seniors, and I'll have to go back and see if I missed anybody. And I really do want to thank you again from the bottom of our heart on behalf of everybody at CSCR. We really do appreciate people taking the time and effort. And as Susan said, I know we didn't get to everybody's questions, and I know not everybody had a chance to speak. I tend to speak too much. And we got the screen up there with Susan acknowledging those who have supported us with some monetary support. You all support us in so many ways, whether you're the parents, supporting your kids being involved in the program, or whether you're teachers. The shout out in closing, that's really, really important, is our connection with the public schools. So I know that there's been a little bit of a change in leadership at the school, and forgive me for not doing justice to all the people in various positions of leadership at the school under Dr. Pat Sullivan, superintendent, and Ed Savage in the science department, chair of the science department. But for everybody who might be in the audience in the or any other school system, we really do appreciate our opportunity to work formally and informally with the area schools. Because as Susan said, the kids are passionate year round, and all of the scientists spoke about the need to continue this research. And we all know in the community that the needs in the research needs in the community don't follow a school calendar. So the more we can keep the kids engaged and work collaboratively in the community for the benefit of all, the better we're all serving each other. So I think that's probably the high note that I will end on. Feel free to stick around and chat if you just want to do that formally. And I'll say goodbye and thank you, and give Susan an opportunity to do the same.
I just want to thank all of you for coming. It's great to see you. It energizes us. And also thank you to our sponsors. We are a very small nonprofit, and we really rely on your support. So thank you very much. And we hope that all of you will think about including us when you think about giving locally. Because we are local, and we can connect you actually in person with the work that's being done. So you can meet some great students as well as see the results of your work. We hope to stay in touch with you. Things are a little up in the air as we look forward due to the public health situation. We keep our eyes on that, and we're trying to maintain our creativity and figure out what we can do to keep things interesting for students and the community as far as learning and researching and being outdoors. So stay in touch. We will have more webinars. Probably our lecture series will come back in a webinar version, at least until we can all get back together in person. And we'll do as much as we can. Let's have a let's all unmute and have a round of applause for the students.
Ryan, wear them. I want to see you.
Wow, I'm exhausted. Hi, Sam. I'm sorry I didn't give you the shout out earlier. Thanks so much for joining us. And Christine Guiney.
Aloha.
It's awesome to see you.
And Lavente and Tucker are three alumni that are on that I noticed.
Yeah.
I just wanted to speak quickly. I don't know if you saw in the chats, and I made a typo, sorry, but great job, guys. This is the type of citizen science that we're looking for in the middle school, the high school. Susan, I worked with you for a number of years with eelgrass, but to see how far this has come since I last left CSCR, which I need to get back to joining again, to see the development of the app, to see the videos, and the amount of students that have come through CSCR is truly impressive. So nice work, guys.
Thanks, Ed. Thanks for being here.
Absolutely. Thanks for having me.
Yeah, we really do appreciate it. I want you to know that.
Can everyone just take a moment to also have a round of applause for Mr. Buckley and Susan Bryant, because they did such a great job at helping us with all of this.
Woo!
I'll clap for Susan. And for Will and Annabelle, and who's still here, and Rosie and Isabel. And Lamenta, I have pictures when those guys were in the 4th grade. or fifth grade. It's so awesome. And for everybody who's in CSCR, you know, if you've been there recently, or a regular customer, in that room, for lack of a better description, the workroom with the pink table in it, the pink room, there's a big poster there on the wall, and it's got the cast of characters. It's got Christine in it, and it's got Clara Baumgarten, it's got Ben, it's got Levente, it's got, I don't know, you guys could probably rattle off some names of other people. And you guys are all about fourth or fifth graders, so maybe a little bit older, but it's fun to see all the people who have been with us and are still with us. And what's our saying, Doc T?
You can check out anytime you like, but you can never leave.
Sure. CSCR is such a lovely place. Such a lovely place.
That is awesome. We really do appreciate the hard work that you did. And please know that there is, for the graduating seniors, there's always a place here for you. And we certainly hope that we can do as much work as we possibly can this summer while remaining safe, and really, as we all want to let our guard down this summer, keeping our guard up, try to do as much work. But if the summer happens to be a summer of a very different flavor, which I think it will be, hopefully you can come back as freshmen in college as interns. So one of our CSCR strategic goals is to actually generate some funding to pay students, so that when they're coming back from college, and obviously the need for a job is even higher when you're a college student, and everybody wants meaningful work. So we really do want to get us into a position where we can have some stipends available for some leadership positions. So hopefully that'll be the thing. And then when the students who are still on, who are the younger students, they'll hopefully grow into a system in which it's a natural. They'll be saying, oh yeah, I want to work at CSCR when I'm a senior or a freshman in college or sophomore in college. So at the risk of saying too much, I am going to cut myself off there. I'm not going to say anything. I'm going to chill out and relax a little bit. I'm going to say thanks so much for all your hard work. You guys rock.
Woo. Yeah. I'm proud of you guys.
Yep.
I kind of have to go. Like, I just have stuff. Like, I have a lot of homework to do.
And thank
on.
Yeah, I did say I was going to be quiet, and see, I couldn't even last 5 seconds. Anybody who needs to go ahead. Feel free.
Thank you.
Thank you.
Bye, Ava.
Bye.
All right. Thank you.
Good job, Andrew and Lucy.
Thank you, everybody. Thank you for spending 2 hours of your day.
Thank you.
Thanks, Andre, for helping to pull together the drifters, too.
Oh my God.
Thank you.
Carl and Andre pulling that together at the last minute. Nice. And Beck. Oh, did he just go? Oh, he just went.
I think Beck's off. Susan, thank you very much. Jeff, thank you. That was terrific. Students, that was fantastic. Way to go.
Hey, Chuck.
Thank you. Yes.
Yeah, see, I can't keep quiet. I have to repeat what I said. The second half of that video was the first time that we saw that. And the second half of the video that we did see prior to today looked nothing like the finished product it turned out to be. What an amazing work effort on the part of Beck to put that all together. So please tell him that everybody really appreciates his work.
It was flawless, flawless. It was beautiful.
You know, the stuff with the arrows, like, you know, here's the beach, boom, 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 boom. Like that thing, that was great.
Forever. All of that stuff takes forever. I don't know how he did it all in the mere number of hours we had since we saw him last.
Well, you know, thanks for giving him this opportunity to and all the students the opportunity to be able to develop these skills. I mean, that this is the kind of research environment that really promotes this type of thing. So thanks, Susan. Thanks, Jack, for everything. It's really rubbing off on everybody. So thank you.
Thank
you. Yeah, you're welcome. Thank you.